So let's today begin by talking about the cell in general, what cell is, how the cell was found, who discovered the cell for the first time, and then we'll go on to the next particular concepts. Let me share you the screen to start with. Okay, let me just go to the first one. Can you see the screen, students?
Yes, sir.
Yes, sir.
Okay.
Yes, sir.
Yeah. So when we talk about the cell, we all know that it is the structural and functional unit of life or living organisms that we call it. So can anybody tell me what do you mean by structural unit of life? Why cell is referred to as structural unit of life?
The cell is referred to as structural unit of life because the whole body is made up of cells.
Whole body, you mean to say in case of multicellular organisms?
Yes, sir.
Okay. Yes, others? Yatish? Why is the cell referred to as the structural unit of life? Try to answer. Other students, you can try to answer. Why structural unit of life? Are you able to hear?
Yes, sir.
Yes.
Others can try. There are 30 plus students. This is a very simple question. We all know that cell is a structural and functional unit of life. तो आपको तो ये पढ़ाए होंगे ना कि स्ट्रक्चरल यूनिट बोलते क्यों है, या फिर आप लोग पूछोगे कभी? Tell me. If you don't know, tell no. I'll just explain that.
Sir, can I tell?
Yes, of course.
Sir, I think at the very root level, the cell is the individual, which is after that we cannot divide it further. An organism is means it is impossible for an organism to survive after an cell.
Survive without a cell, I guess you want to say. What is your name, dear?
Sir, Shaili case.
Okay, please change your name, okay?
Excuse
In
me,
the Zoom app, change the name so that I can understand. Yes, Sahiti.
Okay.
Sir, because all living organisms are made up of cells and every function inside the body is performed by the cells.
So when you say every organism, you talk about multicellular organisms.
Yes, sir.
Okay. So you all are talking about multicellular organisms. So the obvious question is, what about the unicellular organisms? Right? That's a question. So in simple, anybody else want to speak? If not, I will answer that.
Yes, sir.
Yeah, yes, Swagat.
Sir, anything without a cell is not living.
Very good. Anything without a cell is not living. Very good. So all of your answers are right, but again, I would say not 100% correct. So how would I, tell the answer here is, we call the cell a structural unit because structural unit मतलब क्या होगा? The entire organism, whether you talk about multicellular, you talk about unicellular, they are made up of something, a basic unit, a repetitive unit जिसको बोलेंगे. So when we create a building, so when we create a house or a building, or let's say wall, what we do, we take the brick and we just put them together. Number of bricks actually put together repetitively, right? And they're all joined by the cement. Similarly, here also what happens when we talk about organisms, whether you talk about unicellular organism or you talk about multicellular organism, structurally they are all made up of the cell. Yes, in case of unicellular, it is made up of single cell. In case of multicellular, it is more than a single cell. But all of them are made up of this repetitive unit called the cell. And that is why these are called the structural unit of life. Clear on that?
Yes, sir.
Okay. Now the obvious question is, what is why it is called functional unit of life? Now, why functional unit of life?
Sir, I can say.
Sir, many of the mechanisms are controlled by the cells, sir.
Many mechanisms. Okay, many mechanisms are controlled by cell. Again, so of the many mechanisms, some might be useless also. There are many without which the organism can survive. For example, reproduction is not essential for the individual organism. So without that also they can survive, right? Yes, somebody else wanted to speak. Pratik, I guess, or Yatish.
Sir, the cell is told as the functional unit of life because every function of the life is being controlled by the cells.
You are missing out a single word which will improve your answer. You're right. Very good. I'll just tell that.
Sir,
Okay. Yes.
sir,
Yes, Sailikis.
sir, the cell it provides energy which is produced by mitochondria, which
Yes.
is very essential for the living, and all the metabolism activities are controlled by the cells.
Very good. Very good. That's a very good answer. Sai Likesh, I guess. Yes. Anybody else? You should try to answer. There's nothing wrong. Right? Please try to answer. Okay. I guess that's all. Others are not sure about. Okay. Somebody's putting. No need to put. Okay. Nilima. Dear Nilima, please do not put in chat. Sometime I may not be able to see it. Okay. So Nilima is saying functions taking place inside the body of your organism are performed by cells. Okay, that is also correct. Yes, sir. Yes.
Types of functions.
Samiksha, can you repeat yourself?
Which functions which the body.
Okay. Anyways, I could not hear that clearly, but let me tell you that. So we say functional unit. When we say functional unit, all the basic function, basic, the metabolic, the basic, we can say life processes, right? So all the basic life processes that includes the nutrition, the respiration, that is cellular respiration, that gives energy, as some of you said, and also the process of excretion. So all of these essential life processes or basic life processes that are required for an organism to be defined as living or to be in a living state, all of these can be performed by a single cell, right? And in that case, only we can say that is the basic functional unit, because these functions or this metabolism or this life processes are keeping the organism living, and that same thing actually is performed by the cell. So that is why we call the cell to be the structural and functional unit of life or living organism. I hope you all are clear on that part. So when we talk about the discovery of the cell, the first name that comes to our mind is actually Robert Hooke. Okay. And when we say Robert Hooke, we talk about the cork cell that he discovered with a primitive microscope. Remember, at that point of time, the microscope was actually primitive, and whatever he could see actually was limited by the quality of the microscope. So what he saw is the box-like cells. And when we say cork cells, these are dead cells. These are not living cells. Remember, these are dead cells, dead tissue. So they contain the dead cells. Now, what are these cork? Cork are typically present in the mature plants, older plants on the outermost layer. When there is a process called secondary growth, it takes place in a plant. The outermost layer, it forms a structure called cork, basically cork cambium, and this cork cambium gives rise to the cork cells, right? That is part of the anatomy which we will be talking at a later point of time. So this dead cells actually was found or discovered for the first time by Robert Hooke. Give a picture is here. But for the first time, the living cell, right, actually was discovered by another scientist, and he was Anton von Leeuwenhoek, or simply called the A.V. Leeuwenhoek. Now, he saw actually and described a living cell. Now, here's a question for all of you. What was identified? And find out what are the first cell that is actually, first living cell that was actually discovered by A.V. Leeuwenhoek. I want you all to find out. Now, what happened, as I said, the understanding of the cell at that point of time was limited by the quality of the microscope, the standard of the microscope. But over time, what happened, there was a lot of research, there's a lot of improvement in the quality of microscope, including the development of the compound microscope and also the electron microscopes. Now, with this discovery, with this advancement in the field of microscopy, what actually happened is it allowed the scientists to understand, to see through actually inside the cell in a more detailed fashion, and more features of the cell would actually were able to be studied by the scientists. So our understanding today is based on all of these different research that has been done throughout the year. And at the beginning, in the year 1938, right, what happened? There were two scientists, and these two scientists were Schleden, M.J. Schleden, and Theodor Schwann. Now, what these people said is, together, not together, together later on they came up with the cell theory, but independently they formulated or they postulated that the plant cells are composed of plants are composed of number of cells. Okay, Schleden observed this. Now, Schwann also reported that in case of the kind of animal cells, basically reported that thin outer layer that is called a plasma membrane is actually present, and he also found that there is an additional cell wall that is actually found in the plant cells. And he proposed, Schwann proposed, that both the plant cells and animal cells are composed of cells and they are a product of cell. Okay, I request somebody to switch off the microphone, and I'll just do that. Please switch off the microphones. Okay. But what happened was they have explained many of the factors, right? Schleden and Schwann proposed many things, right? They said that all organisms, like you talk about plants or animals, they're composed of cells and they're product of the cell itself, right? And how the organism is going to behave, it is based on the organization of the cell. The cell's behavior is going to affect the organism's behavior. So many of these theories were put forward by Schleden and Schwann, and they originally formulated a cell theory. Now, there was a problem, there is shortcoming of the cell theory proposed by these two people. Now, that is what they could not propose how the new cells are forming or developing. So this particular question, what is the origin of the new cells? Now, that particular question was not answered by these two scientists. Now, Rudolf Virchow, another scientist in the year 1855, now what he did, he explained the concept on the origin of new cell. And what he said is the new cells are going to form as a result from the pre-existing cells. That is omnis cellula acellula, that is new cells derived from pre-existing cells. And he modified the cell theory, and the cell theory today stands with two statements. We can understand that. What is the statement of the cell theory? Now, first point, all the living organisms are composed of cell and the products of cell. Now, this comes from the Schleden and Schwann's observations, and it was modified with the second statement by Virchow, that is, all the cells are arising or is derived from pre-existing cells. Now, these are the two theories, or these two statements are part of the cell theory that you all have to understand. Are you able to understand, students?
Yes, sir.
Yes, sir.
Yes, sir.
Okay, let's move on to the next part.
Yes,
An overview of the cell. And I guess, and I'm pretty confident that you all are going to have a pretty good idea about what I'm going to speak now. Because every cell consists of certain components. One of that is going to be the cytoplasm. Now, what is cytoplasm? It is the fluid component, or it is a semi-fluid matrix, right? What is a matrix? Matrix, as we talked about earlier, it is a, it's something that is going to bind the cells. If you remember in the cell chapter, structural organization in animal tissue, we had talked about the matrix. So here it is a semi-fluid matrix where all the cellular activities most of the cellular activities are actually performed, and number of these metabolic reactions or chemical reactions are actually going to take place. And remember, the cytoplasm contains a number of the organelle, right, that we know about. They contain that, they provide a space for this organelle to perform all of their functions, right? And this basically keeps the cells in a living state. Now, what about ribosome? Because ribosome is very essential and they're present in almost every cell, actually in every cell. and these are not membrane bound, right? So ribosomes, we'll talk later, are consisting of something called ribosomal rRNA and number of proteins. So this protein and the ribosomal rRNAs or ribosomal RNAs or rRNAs, they form the structure ribosomes, and remember, they are not going to have any sort of membrane, right? And they're present within the cytoplasm and also in chloroplast, mitochondria, right? Apart from cytoplasm, in the chloroplast and mitochondria also they are found, and also on the RER or the rough endoplasmic reticulum. Now you all might be knowing they play a very important role in the process of protein synthesis, right? So they play a very important role. Without these structures, without ribosomes, proteins, which are essential, very much important biomolecule in the organism, cannot be synthesized. So they are very important. They are present in the cytoplasm. Along with that, they're present in chloroplast, mitochondria, and also on the surface of the endoplasmic reticulum. So when we talk about the different shapes and size, there's a lot of variations, right? Morphologically, they're very different. Structurally, they're very different. Because why are they so different? Because of the adaptations for the roles they are designed to perform. So every cell is going to perform a specific task, specific role. And in multicellular organism, there is a division of labor. I think this term, again, you might be knowing very well. There is something called division of labor, right? So whatever work is there, let the work be divided among all the cells in a multicellular organism. All the cells are different type of cells in a multicellular organism. To give you an idea of that, RBCs, red blood cells, specific role, leukocytes, specific roles, right? Then you have the platelets, specific role, you have the neurons, a specific role, right? And then you have the fat cells, there's a different role for it. So every cell is having unique role. Muscle fiber is, for example, is going to have a different structure with different components for a particular role of contraction and relaxation. Now imagine the muscle cells being very small, just like the RBC, and the neural cell is also just like, let's say, the RBC. And imagine the RBC to be elongated and to be branched like the neurons. What is going to happen? In that case, the blood cannot carry the RBC if it is in the shape of a neuron throughout the body. You would have heard about a condition called sickle cell anemia. How many of you have heard of that? Sickle cell anemia का नाम कितने लोग सुने हो? Anemia, sickling we call it, right? How many of you have heard of that? Have you heard or not?
Anemia heard, sir.
Sorry,
Yes,
Samiksha?
sir. Yes, sir. Anemia heard, sir. Word.
Yeah, okay, good. So in case of anemia, I think others may not have. But what happens? There is a loss of blood, and because of a single mutation, what happens? Your entire RBC's shape from a typical biconcave changes to a crescent shape, or it's like half moon shape. Because of that, there's a lot of problem in carrying the blood, or basically within the the blood capillaries or blood vessels. They get stuck, and they might cause lots of problem, and in serious cases, it might even lead to cardiac problems. So if the cell shape, what is currently is there, if changes a little bit also from a biconcave to crescent shape, you can see a lot of problems are there. Now imagine if the shape is completely different, just like a neuron, it is going to be disastrous, right? So what I'm trying to say is every cell in our body is having a unique shape, it is having a unique structure, and that unique shape and structure allows the cell to perform a specific unique function that is designed to do. So when we talk about the shape of the cell, that also matters, right? Whether you talk about unicellular or multicellular organism, the shape of the cell and the size of the cell is also going to be a very important aspect for the existence of that particular organism or the cell in the multicellular organism. So when we talk about the smallest cell, we talk about the mycoplasma, right, which is going to be around 0.3 micrometer in the length, right? You have learned about this in the previous chapters also. So when we talk about the largest isolated single cell, that is going to be the egg of the ostrich, a class 8 concept that you all must know. Longest cells in human body, that is going to be your nerve cells, and the size of the bacteria is going to be around 3 to 5 micrometer. And the human RBC, right, human red blood cells, they're going to be around 7 micrometer in diameter. And as you can see, all of these have different shape also. You can see the bacteria having different shape, right? And RBC having different shape. Understood. Now, based on the function, shape of the cells may be disc-like, polygonal, columnar, cuboid, thread-like, or irregular, both in case of plants and animals. We're not talking about only plants or only animals. Both in case of plants and animals, depending on the function that I was talking about, there is specific role, right? So you have the parenchyma in one particular organization, collenchyma is there, sclerenchyma is there, and we're talking about only these three because these simple permanent tissue, they organize themselves in various combinations to give rise to the complex permanent tissue that is going to be xylem and the phloem. And the other type you all know that is going to be meristems, right? So these have a specific organization because of the roles that they're going to perform. Similarly, when we talk about nerve cells, muscle cells, bone cells, right, you have the gland cells, glandular cells, then you have the blood cells, different type of blood cells, and sperm cell and the egg cells. Each of them are going to be having its own role or function based on the structure that they're having. So you know different type of structures here, as you can see here. I try imagining that if we change the cell shape to something else, we keep exchanging the cell shape, what is going to happen? Can they function? Try to imagine that. Now, based on all of the other, in a general features and differences, we can say the cells are of two types. And that's, my dear students, is very important. We have to understand that. And that is called the prokaryotic and the eukaryotic cells. And to start with, I guess many of you might have a basic idea of that, but I'm going to show you a very, interesting animation of that. And then we'll continue to the next part. Okay, just have a look, and this let me just enable share computer sound. Yeah, I hope you can see. We can now.
All living things are made up of cells. There are many different types of cells in our bodies, including bone cells, cartilage cells,
Yes, sir.
Yes, sir.
Yes, sir.
cells, and nerve cells. The broadest classification of cells is into two groups, eukaryotic and prokaryotic. There are a number of differences between these two types of cells. The main difference is that eukaryotic cells have a double membrane-bound nucleus, which contains the cell's DNA. Prokaryotic cells do not have a nucleus, only a nucleoid, which is the central, open part of the cell where the DNA is found. Eukaryotic cells also have other large, complex, membrane-bound organelles, which prokaryotic cells lack. These include mitochondria, rough and smooth endoplasmic reticulum, the Golgi complex, and in the case of plant cells, chloroplasts. Organisms with eukaryotic cells are called eukaryotes, and they include all animals, plants, protozoa, and fungi. Organisms with prokaryotic cells are called prokaryotes, and they include bacteria and archaea. For millions of years, prokaryotes were the only form of life on this planet. Eukaryotes came later as a result of the process of evolution. Another difference between eukaryotic and prokaryotic cells is their size. Eukaryotic cells are generally larger than prokaryotic cells. Eukaryotes are mostly, although not entirely, multicellular organisms, whereas prokaryotes are always single-celled or unicellular organisms. Examples of unicellular eukaryotes include amoebas, paramecium, and yeast. The structure of the DNA in eukaryotic cells is different from that in prokaryotic cells. In the nucleus of eukaryotic cells, DNA forms tightly bound and organized chromosomes. Prokaryotic cells contain just a single loop of stable chromosomal DNA stored in the nucleoid. The nucleoid is not a structure, but the area where the DNA is found. Both types contain ribosomes, but in eukaryotic cells, they are bigger and more complex and bound by a membrane. Most eukaryotes reproduce sexually. The offspring have genetic material that is a combination of the parent's genome. Prokaryotes, however, reproduce asexually. Their offspring are clones of the parent cell, which come about through binary fission. Finally, prokaryotic cells have a larger surface area to volume ratio than eukaryotic cells, which results in a higher metabolic rate and therefore increased growth rate and shorter generation time. While eukaryotic and prokaryotic cells are quite different in their structure and processes, they do share similarities. Ribosomes are one feature they have in common, but both also have a cell membrane composed of phospholipids and proteins. The membrane provides a barrier between the external and internal environments of the cell and selectively allows certain materials to pass through. Both types of cell have DNA as the basis for their genes, although the structure is different. The genetic material regulates cell function and contains the coded information that is passed on to offspring. Both also contain cytoplasm, but in eukaryotic cells, it is defined as everything within the cell outside of the nucleus. In prokaryotic cells, the cytoplasm refers to everything contained inside the cell membrane. The gel-like cytosol is a major part of the cytoplasm in both types of cell. This solution is the site of many of the cell's metabolic processes, such as the synthesis of protein.
Okay, so I hope you have a very good idea now regarding this particular structures, right? Agree or not? What is about the prokaryotic and eukaryotic cells? Did you like the video?
Yes, sir.
Yes.
Yes, sir.
Okay, I'm sorry for that. Lot of bats nowadays. Anyway, so with that in mind, you have seen the basic idea that both the prokaryotic and eukaryotic cells, they share certain similarities, but mostly they're different in many aspects, right? From the organelles, right from the the organization of the nucleus or the genetic material, and also even the difference in the cell size, and also there is a difference in the ribosomal types. So we'll talk about all of these in the subsequent slides. So let's start with some basics now, and then we'll go on to the details at a later point of time. So when we talk about the prokaryotic cells, the first and foremost thing that you have to understand is we are talking about mostly the bacteria, right? So the bacteria are actually that we have learned already is actually all the prokaryotes, and they have no membrane bound nucleus or organelles. Here they're all naked, we can say it. As we will learn, there is a structure called nucleoid, but this nucleoid is there is an area called nucleoid, but nucleoid is not a structure, it is not an organelle where the nucleus or where the genetic materials are present. It's an area where the DNA gets combined with the protein, right? DNA is, we'll talk about it later, the DNA is actually negatively charged, and this negatively charged DNA combines with proteins which are positively charged, and they form the structure called nucleoid. Yeah, it is present. Now, these prokaryotic cells, when we talk about, they include the bacteria, right? If you remember, we talked about two categories of bacteria, archaebacteria and eubacteria. I hope you remember that. But anyway, bacteria are there. Then the blue-green algae, or the BGA, or it is also known as the cyanobacteria. I hope you remember. Then we have the mycoplasma and the PPLO, pneumonia-like organisms. So all of these are going to be the prokaryotic organisms or prokaryotic cells. And typically they're smaller than the eukaryotes, and they can multiply much faster than the eukaryotes. Now, to give an example, the bacteria, it takes only 20 minutes as the generation time. 20 minutes as generation time. That is, within 20 minutes, it can basically copy itself. The new organisms, one generation basically takes only 20 minutes. So around 18 minutes takes for the nucleus division, and the remaining time is spent for the cytoplasmic division. And then you have a new organism within 20 minutes only. Why? Because these organisms are not very complex. They have a very simple organization, and that allows them to reproduce in a shorter period of time in a simple manner. And of course, they vary a lot in the shape and the size. I hope you remember the different shape, right, of the bacteria also that we had talked about. If you remember, the bacillus, that is the rod-like structures are there, then spherical, coccus, vivarium or column, comma shape, and you have the spilum or the spiral shape, as you can see here, bacillus, coccus, vivarium, and spilum. So there is a variation in the cell shape, just like what we are talking about in the multicellular or eukaryotic organisms. In that also, there's a lot of variations. So in general, if we talk about the prokaryotic cells, cell organelles, we'll talk about that. But anyway, we have the cell envelope. Yeah, then we have the mesosomes and chromatophores. You have structures that is called nucleoid, or a region called nucleoid, flagella, pilion fimbriae, ribosomes, and inclusion bodies. And we will talk about each of these from the in detail from the next class onwards. So anyway, just to have a basic idea, you can see this is the area where you have the DNA, right? And this DNA, as I was talking about, is going to be negatively charged, right? Negatively charged, why? Can anybody tell me why DNAs are negatively charged? Any idea? Yes or no? Why are DNA negatively? Okay. So we'll talk about the DNA structure, but just basic idea you remember, DNA contains phosphorus group. And because this phosphorus group, right, phosphorus group or phosphorus moiety, the DNA is actually negatively charged, right? And the proteins which are associated, they're actually positively charged. Now remember, opposite charge attracts. So this DNA negatively charged combines the protein which are positively charged, and they organize themselves into a region that we are calling is the nucleoid. Okay, it is not membrane bound, simply association of negatively charged DNA with positively charged protein. Now, apart from that, what happens? There is an additional structure also which is not mentioned here, which you all must know, and those are a double membraned, double-stranded circular DNA, and this extra chromosomal double-stranded circular DNA, which is, which has the ability of cell replication, they are called the plasmid. And we'll talk about this, which is very important that you have to understand this, because this plasmid allows the cell, this bacterial cell, to have certain unique feature, something that is, not possible without the presence of the plasmid cells. And sometimes you might be knowing that the bacteria can survive against antibiotics, right? We typically use antibiotics to kill the microbe, but sometimes antibiotic does not function, or you would have heard about multi-drug resistance or antibiotic resistance. I don't know, some of you might would have heard. So that's because if the bacteria somehow acquires these plasmids, there are different types, we'll talk about that, but in general, if they can acquire this plasmid, they can have the ability to resist the damage by these antibiotics. So that will allow them to live just within poison, right? Antibiotics are like poison for them, but they can survive if they have this plasmid. So this extra chromosomal outside the regular DNA, outside the chromosomal DNA, there is this additional set of DNA which is double stranded, and that, dear students, is going to be referred to as plasmid. So we'll talk about these structures as it's written on the screen in detail from the next class. Will continue. So that's all for today. Thank you all, and I hope that you have understood these concepts and the class was interesting for you all.